Got it recording.
Okay. All right. First of all, welcome again, and thanks for attending session 2 and 3 of the training sessions today. And through this presentation, we'll just be discussing the use of antibiotics, why we're using them, how to prepare them, and how to apply them when we're out in the field. So without further ado, I'll get straight into it. So as Dr. McCoy said from the previous presentation, SCTLD is a highly virulent disease. It's very transmissible, spreading from coral to coral very quickly. And it can also result in mortality very quickly as well. In some cases, it depends on the coral species, but it can travel over the coral at a rate of 1 to 3 centimeters per day, which is pretty quick. If you consider you've got a 10 centimeter big coral over the course of that week, if you come back, it'll probably be dead. So this is a pretty serious disease that we're dealing with. So a question that is on everybody's mind is why we're using antibiotics. Well, there's actually a couple of reasons for this. One reason is because other natural non-antibiotic methods have been tried and tested, but they just haven't had the same success rates as using antibiotics. It's very easy to administer underwater by the scuba divers. After a bit of training and using them day in, day out, we become very proficient at applying the treatments to the corals, which is very good. Hopefully you guys will be just as proficient when you come up with us as well. And just adding to the fact, there's been a lot of scientific studies that have been peer-reviewed by other scientists in the region of the Caribbean who have also used amoxicillin and this special paste, what Croy mentioned before, and it's been really effective in halting the lesions on corals that become infected. So just carrying on from that, there's been a lot of studies in the past that have used alternative methods, such as not doing anything at all. As you can see there, this resulted in a 97% mortality rate. So out of 100 corals, doing nothing led to 97 out of 100 corals dying. Some created a physical barrier with some tools. That also was not too great. The chlorinated barrier didn't also work either. But what we did see was with using the antibiotics with the base 2B, yielded quite positive results. So this was then agreed by a few countries, like in Florida, that we should be using antibiotics at the moment to treat infected corals. So a couple of things to think about whilst we use these antibiotics is, although the treatment, is doing a good job at stopping the progression of the lesions on the corals, it doesn't stop new lesions from popping up over time. So you may treat one coral with 2 lesions 1 week, but you may come back in a month's time and there's 3 new lesions on top of the ones you've just treated. So it doesn't stop that from happening. Again, it's just like humans. When we take antibiotic supplements or treatments from our doctor, it normally requires like a periodic retake of the antibiotics to get rid of a infection or something like that. It's the same for corals as well in general. When we do treat the corals, we can go back and revisit the corals and just put another treatment on the coral, but it usually doesn't take as much antibiotic as the first time that we treated it. The treatment success, it does vary from species to species. we know that during our study, we've seen a lot of success with studies in Florida and around the Caribbean. They've all been showing different success rates, but in general, it is the treatment that is working right now, and just due to the severity of the disease and how fast it moves across like Grand Cayman's reefs and stuff, like for now, it's the best for us. So what have we seen so far? So right now, during our project, we are actually involved in a study where we've tagged 47 different coral colonies from 8 different species at Lighthouse Point dive site, which is where Dive Tech is based. And if you've been diving there recently on a fun dive, you may have seen some corals on top of the hard pan by the boat moorings, which are tagged. And this experiment is just to see or ascertain how effective the antibiotic treatment is that other people are using around the Caribbean region. And in due course, we will be adding our results to the breadth of literature that is out there already. So what we've been seeing so far is, as time has went on, we've noticed that the corals seem to require less and less treatments, which is really good. As you can see, in June, they were all infected, which is what we wanted, because we needed to then treat the coral, to then assess it week after week if they got a new lesion or whether the treatment halted that lesion. And then as we come into August, we start to see a response to the treatment over the whole number of corals that we have. And then as we got to November, as of last week, 70% of the corals are now stable, which is really positive news for us. So even though we are using antibiotics, there have been a lot of concerns that are being raised by different environmental groups or individuals, and rightly so as well. But we thought we would try and cover some of the concerns that might be raised here by you guys or other people who have done this training as well. So the first concern is obviously, what if there are instances where the microbial community are on corals become resistant to the antibiotics that we're using? And of course, this happens all the time. It happens with humans as well. Like, we take antibiotics all the time. Viruses mutate as well, and microbes are all different themselves, and they're very complicated. But a study, I think it was like last year, it found that there was a resistant gene in 2 out of 24 samples that were taken from corals. But what we really need here is like a larger sample size to further try and nail down if this is actually the case or not, whether this, we find this in a lot of different regions where we're using antibiotics. I guess the positive thing is that we are using particularly high dosages within this, the treatment. I think you may come across this problem with antibiotic resistance if you only are using a small dose of antibiotics, like frequently over time. Like, we know just from Florida that this disease has been around for 7 years now. But we are hoping to explore other alternative treatments as well. So we're hoping like it won't lead to that. But for right now, we think it isn't. It's of little concern right now. Another point is about antibiotics entering the food web. This has been happening for a long time now. As I say, humans have been using antibiotics for many decades, and they're in the food web already. But what is different about this is that corals themselves aren't a particular major food source for a lot of animals, only for like the butterfly fish and And sometimes, you know, the parrotfish do chomp and bite at the corals, but they're wanting the algae. They're not actually wanting the coral polyps like the butterfly fish. And although this can be explored, we aren't doing any research about that. But there are people in Jamaica who are going to be assessing the impact of the treatments on certain reef fish species, probably the butterfly fish, because that's who we associate this disease with. So that is a concern, but we'll see what that study comes up with in time. Another concern is usually how much antibiotic are we introducing to the marine environment? Well, all we can say is in comparison to the natural background levels, like it's quite minuscule around like developed areas. Around Cayman, we don't have that much like treatment waste or anything like that. So for here, we're in quite a good position, really, that we're not, you know, introducing too much where it's going to raise levels where it is going to be of concern. But for areas like Florida, where there's a lot of agriculture, aquaculture, just you know, treatments from waste and stuff like that, that enters the environment there quite a lot. So when you compare this to us taking 20 syringes out with 200 grams of amoxicillin being essentially put into the environment, it's really minuscule in comparison to these other activities that go on around the world. And I think we're in a good position here. Not that we should be doing it, but for right now, this is like the best thing for the corals. So what is the treatment actually made up of, and you know, how does it work? Well, there's two parts to the treatment. It's quite simple. One part is a hydrophobic paste which adheres or sticks to the corals very well. This is specially made by a company called Ocean Alchemist, and they're based in America. We use amoxicillin trihydrate as the antibiotic. It's a broad spectrum antibiotic, and it treats a lot of gram-positive and gram-negative bacteria. And it's mixed together in an 8-to-1 ratio. So 8 parts of the paste go with 1 part of amoxicillin. So the treatment's designed to stick to corals specifically, like this is what we're using it for. As you see the picture on the left here, this is an infected coral with SCTLD, and you can easily see the boundary line between the diseased tissue and obviously the healthy tissue just on the other side. And we basically just put that on the boundary with the syringe, which we'll talk about in a few slides. And this is what we do when we're underwater. But once it's in place, when we've put this paste on, the paste actually, as Croy was saying before, it controls the release of the amoxicillin into the coral, into the tissue where the infection is happening. And this happens for about 72 hours or so after it's been applied. When it's been 3 days, it rapidly disintegrates into the water column whenever it's exposed to water. But it's quite localized, and the amoxicillin stays within the paste. That's not to say it can come off with currents or fish, but most of the time we'll come back and we'll see the picture like here, where we can see where we've treated it before. And there's some residue of an old treatment here. And you can see it's done a pretty good job of halting the lesion. Obviously here is a break where, you know, some treatments came off, but that could have been after it's inactivated after that 72 hours. And this is a symmetrical brain coral for anyone that's interested, which we treat a lot. So when we are preparing the treatment, we've got to ask the volunteers, you guys, we've got to ask if you're allergic to amoxicillin or anything like that, because obviously we're using this in the lab, and if you accidentally breathe it in or get it on you, then this can result in a hospital visit for you guys. So unfortunately, if anyone is allergic to it, they do need to let us know before they come in, and before they join the team and start mixing the antibiotics. So if you are, do let us know. And if you are, then unfortunately you will not be able to participate in just this aspect of the project. Obviously, this is a major health hazard and a lot of paperwork for us. So sorry. But if you aren't, then we can continue. Only joking. So to prepare the treatment, we have the base 2B, which we have to store in the fridge. It keeps it a nice consistency. Otherwise, if it gets too warm, it becomes less viscous and less sticky. So that's kept in the fridge or in a large ice chest that we have just full of ice. We replenish the ice every couple of days as needed. We have the amoxicillin, which is stored in a large container in one of the labs under a fume cupboard. And that can be stored at room temperature. So like less than 25 degrees Celsius is okay. So just for an example, if when we're preparing this, for one syringe, well, that should say 60 milliliters. Sorry, that's a typo. That's my fault. So you just need around 70 milliliters per syringe that we have. And this goes with about 9 grams of amoxicillin. And then as you're preparing more, then you can just look at a cheat sheet, and then you can double it if you need 2, or if you need 10, times it by 10, and you'll get your amount. But the SCTLD team members will always be with you when we're going to be mixing, so they'll have the numbers covered, hopefully. But it does vary day by day. Sometimes we need more, sometimes we need less. Sometimes we have some left over from a previous day's treatment. That's okay. It'll be good for the next couple of days. As long as it's just not in contact with water, it only degrades at a rate of about 1.5% per day in terms of effectiveness. So that's pretty good for us. So this is just a guide for the preparation. Whenever you come up to help us, you will be involved in this this task in the morning or in the evening if we need to make a lot for the next day. So you're just going to measure out the desired amount of the base 2B. You're going to do this for your amoxicillin as well, but you're going to make sure you are working under the fume hood for step 2 with the amoxicillin, because sometimes it's quite puffy and it can go all over the place and you don't want to be breathing that in. But just before you do this, just make sure you're wearing a mask before opening it or dispensing it. Next, you're just going to slowly and carefully pour out the amount you want into the mixing bowl. We have a big fancy mixer now, and yeah, we've upgraded our methodology quite a bit since we first started about 6 months ago. And you're going to want to run this for about 5 to 10 minutes, get a nice smooth consistency in the treatment so that it can be packed into the syringes quite easily. And what we normally do is add a non-toxic food coloring to the paste. It just gives it a nice aesthetic look, and you can see when corals have been recently treated when we're on the reef according to the color. But it all turns back to a normal beige color after about a week or so. Step 5, if you've got a large batch, then you're just going to repeat steps 3 and 4 because it's just easier on the mixer. The equipment's not cheap to buy, so just so we're not overworking it, we're just going to we'll sometimes split the load into 2 and just repeat it. It's all good. Or as you can see here, we've got Scott and Joe, some of our SCTLD response team members that are having, I think, way too much fun, packing the syringes and putting the antibiotic into it. They like it, and I'm sure you will when you start as well. And finally, just when we've packed all the syringes, we literally just put them into a cooler box for transportation. That goes onto the boat. It's as simple as that. So all that's prepared. We want to now talk about, before we actually go in the water, what we're going to need. So what you're going to do first is just make sure you listen to one of the dive captains or one of the dive team who will give you a briefing on what we're going to be doing that day, where we're going to be going, what we're going to be doing. Sorry, repeated that. And where we're not going to be going, especially when we're in the water. And just a few things to note is like the current and stuff like that. Like this is going to influence, your attack on the area. You don't want to go too far if you're down current, because you've got to come all the way back to the boat. And just be wary of like the distance you travel as well. Like if you're not that fit or healthy, things do happen and you can get into trouble. Just make sure like you're quite close to the boat. And even before jumping in the water, like every buddy team should have, basic equipment, like, for the syringes that you're going to be using to treat the corals. You're going to need a camera for that as well. At least a camera each if there's not that many members on the boat, people, sorry. And you're going to need a scale bar as well. So this is it, just in picture form, but it just gets your mind jogging. And, there has been instances before where we've all forgot stuff when we get out there, and that can make the day drag a little bit longer if we need to go back to the dock. If someone's forgot like the gloves or scale bars or cameras aren't working, we have to check that we've got all these things and then that all these things are working properly. Really easy. Something that we do have to take into consideration is when we get down there, like what corals are we going for? Is it just any coral that looks like it's got a disease that we treat? And the answer to this question is multifaceted. On one hand, you can go down there and it might just be a small area that's infected. And like for that instance, I'd probably say, try and treat every coral that you see. But if not, if the area is vast and there's hundreds of corals that need treating, then we do normally have a list of like corals that we are highly susceptible and do need a lot of treatments. But when we're actually coming up to the coral itself, we want to be assessing it on two factors. One of them is if 50% of the colony is still alive. If it's more than that, then we don't really want to be, well, it's not wasting the product on it, but because it's, we've only got a finite amount of this treatment that we have, it's advisable to just go, if you see a coral that's, more than 50% healthy, treat that coral instead. Another good thing to think about is just see how many lesions the infected coral has. If there's more than, if there's less than 5 lesions, then you can probably infer that this coral here is more healthier than the coral that's got multiple lesions on it. So it's this coral is obviously less fitter than this coral. So we'll prioritize the treatment on corals that have just got a few lesions, but sometimes this isn't possible. And again, it's your judgment, because you may not always like be right beside your buddy who will be a SCTLD team member who can advise you. In a case where you're by yourself, but your buddy's not really there, just make a judgment based off this, based off these factors. And also, I think a third reason would be if the colony's been treated before, if it looks like the disease has broken past the disease barrier, the treatment barrier, sorry, you may just want to touch it up with a little bit more treatment. In the long run, it doesn't use that much more, and it gives the coral like a better chance at surviving like that particular lesion, which will be good. And also not mentioned in here is I would also, when you're looking at an infected coral, just if you've got like a large coral, that's like say a meter long, you know, 50% may be dead, but that 50% that's alive is still a lot bigger than a lot of the other corals that are in that area. So what I would do was probably focus my treatment on that larger coral. And the reason behind this is because the larger corals have greater reproductive success. So they're able to produce more eggs or sperm whenever it comes to spawning, which is the end of September for most species, full moon. So that's just another hint as what you should be treating when you're down there. Okay, so you've seen your infected colony that you want to treat. What do you do next? Well, first of all, you've identified it, so now you just want to put your scale bar down, which every person will have, and which is this object right here. This is just so we can get a picture of how big the actual coral is that you're treating. What you want to do is just make sure you're placing it on the ground, just in front of the coral. And obviously, if this is not possible and you're on a wall dive and the coral's on the wall, just literally place it in front of the coral or just below it. But by no means don't ever put it on the coral itself. That's because we don't want to risk the transmission of disease if it's on, say, the scale bar itself. We don't want to be adding to the problem. So when you're ready to take your picture, just make sure you've got the coral and the scale bar in the frame. Once you've got that, you're good to go. Pretty simple. Now, when you've got your picture, you put your camera away, and you put your scale bar away, and you want to just apply a band of paste. You can't really see it here, but on the next slide, there'll be a video. You just want to put it across that boundary, but between the healthy tissue and the dead, recently exposed coral skeleton. And you want to do that because when you push it down onto the skeleton part, it will act as like an anchor, and it sort of holds it in. Because obviously on the live coral part, it's going to be producing mucus and stuff like that, and it makes it really difficult to adhere to it. Okay. And just make sure it's an even band of treatment that you put across the boundary. If there are any thinned out areas, just apply a bit more. It's all good. Just apply as and when needed. And when you've treated the coral, just take a picture when you're done, just one will do, of that treated coral. So this is just a video of me on a coral that I'm treating, which is diseased. Here you can just see, it's not a very great video, but you can just see the boundary between the healthy and diseased coral here. And I'm about to apply the treatment. See here, I'm taking very good care of my buoyancy, making sure I'm trying not to touch stuff, but obviously that sea fan's just in the way a bit for me. But I'm trying not to touch any other corals just to limit transmission spread of the disease. And then you can see here, I've seen another area that needs treating on the same colony. And now I'm just going to take a picture when I'm finished. So just a couple of easy steps. And when you're in the field, you obviously get a better picture of what we're doing, and we'll be there to help you to make sure you're doing it correctly. And, as I'm talking about this, I might as well carry on with a few more tips. Some corals are easier to treat than others. Like the coral I was treating right then, it's quite easy to treat. It's one of the star corals. But if you see in the field, you may encounter some other corals, especially the mountainous star coral. Oh no, sorry, the great star coral, which I can't point out right now because I haven't got a picture for it, but it was on one of the pictures in the other slide, so you can look at this back and see that. It produces a lot of mucus sometimes, and it's very hard sometimes to adhere the pace to it. But it's, you know, it's practice makes perfect, and you just get used to how you do it, and you get better. That's just how it works. Obviously, if you're working in shallow waters, be aware of surge. You know, you're going to be up and down in the water column, and you know, you might, you know, dispense a bit of the antibiotic by accident if you like try and hold on to something, or you know, just be aware of the conditions at the time. Just wait for a lull in the surge. Like I said before, practice makes perfect. We do have different ways of doing everything from, you know, in day-to-day life, but these are kind of the instructions that we've been following from experts in the field who have done it before, and you know, these are all proven to work. So we're just building on this and passing it on to new volunteers like you. So when you're applying the treatment, just make sure that you're not actually physically holding the coral that you're treating, because obviously, again, you don't want to be transmitting any disease that may be on your gloves, as we use them all the time. You know, it can happen in times of strong surge that you'll be bumped into something, but sometimes it can't be helped. But if you can, just please don't be touching the coral anyway, as it, you know, it's against the law here to do that, but we've got special permission to treat these corals, but we don't touch them in any other way. We get that permission from the National Conservation Council. Just be aware of that. A good thing to note is the syringes, when they're full, they are positively buoyant, so they will float up if you let go of them. So just make sure you store them in your bag, the ones that you're not using, and then put them in your pocket, or if you've got a bag attached to the BCD, keep them in there, because if you let go, they're going to the surface and we probably won't find them again. So just make sure you're holding on to them all the time. And just to end, you know, we talked about antibiotics, and it is the treatment right now that works for us, and it's great. It really works. But obviously, you know, we don't want to keep putting antibiotics into the environment as well. You know, we are advocates as well of keeping it pristine and as clean as possible. But there are some promising future alternatives that very clever people are working on, like in the Smithsonian Marine Institute in Florida. They're working on a probiotic treatment. We've actually been sending samples from the Cayman Islands corals, where we just take little tiny pieces from healthy corals, diseased corals, and corals that have been treated with antibiotics. And the idea behind this is to see if there's any changes in the microbial community that lives on the surface of these corals inside the mucus, which is on top of the coral colonies. And hopefully from this, they will try and isolate and culture positive, strong bacteria that are able to like be resistant to SCTLD as a disease. And this research is still ongoing, but they're making very good progress, and we expect to hear more positive news soon. So if you want any more information about that, then you can email us, and then we'll disseminate some reports that we know are available. So you've got that information, which is all interesting reading. And instead of using antibiotics, we'll use these probiotics to treat infected coral colonies. Like you can just see here, it basically has like a weighted mesh bag that surrounds the coral, and then the probiotics are pumped inside the bag, and then we wait a few days, and there have been very good results that have came out of some pilot studies regarding this research, which which is very good for us, and we'll hopefully be wanting to take part in this as well. And another thing is Ocean Alchemists, who, as you know, they produce the base 2B that we get. They're working on something themselves by using antibacterial products to kill these gram-positive and negative bacteria. I mean, this is what the amoxicillin is designed to do as well. But again, we don't want to keep using antibiotics in the marine environment, as I said before. So hopefully Ocean Alchemists can further their research in that and study and let us know in due time. If they're successful, they'll be holding trials, and then there's quite a long process before we can use it here. So it's all very promising. But what this allows us then to do is we don't have to mix it ourselves, which, saves us time, and we can use that time to do other stuff. And I guess another benefit here is that it will have a longer shelf life as well, so we can just store it in the fridge as well. Yeah, it's all looking very positive, and yeah, we're looking forward to going forward in the project, trying to find alternatives. But as I say, just for now, the reason for using antibiotics is because it really works. And if there was something else right now that was 100%, then we would probably switch. But yeah, it's going quite well, and we're quite happy with the results we're seeing. And obviously around the Caribbean as well, people are also seeing positive results, and the research is still ongoing, and hopefully we can contribute this in due time. But from me, this is the end, and if you've got any questions at all, you can direct them to me and Croy. Yeah, thank you very much for listening.
Yeah, great job. Thanks a lot.
One more question. You mentioned how it's not an infinite supply of the treatment. So how often do you replenish your stocks of it, and how much do you get through in a given period of time, say a year or 6 months?
Yeah, that's a good question. Okay. Yeah, it's not an infinite supply. We normally get, the last shipment we just got was for 25 kilograms of amoxicillin, and this we actually sourced from India, and it's the same amoxicillin that we've been getting from America, but it's just at a greatly reduced cost. I'd say per day we use we probably go through like 40 syringes, and say in each syringe there's like 9 grams of amoxicillin. It's every couple of months, every 2 months maybe, we would replenish our stocks. But for the base 2B, we obviously need like a lot of that. So we normally order like 25 1 kilogram tubs at a time, and that lasts us about 2 months, I think, Croy, something like that.
Yeah, with 2 strike teams, we with Easton and and us, it lasts us about a month.
Yeah,
it's not cheap.
No. we're working on it, and yeah, we're learning as we're going through the project.
Can you not substitute the base 2B for any marine epoxy, like the coral creep putties, or what the things that they use for coral outplanting? That's
a great question. There have been studies where they've used epoxy as a substitution for the Corex base 2B, but the results just aren't as good as the base 2B at halting the disease lesions. And the epoxy is quite expensive in itself as well, and that's in limited quantities as well, being on an island. Like this paste is specifically designed to adhere to corals like quite well, and it does the job quite well. But there have been studies, but they've just not been as successful as halting the lesions, unfortunately. whether it's due to the chemical composition of the clay or epoxy or putty, it just doesn't hold its own against the H2B. So hope that answers your question.
Yeah, absolutely. Thanks.
Just to add to that, the base to be, as I understand from Katie at Ocean Alchemist, the fact that she used her tongue as what would stick to that, because it's very difficult for anything to adhere to the mucus layer that corals have underwater. But we have tried epoxy, chlorinated epoxy, you name it. Different researchers have tried with little or no success outside of using this specifically developed for that, infused with amoxicillin. So that's what the whole region is pretty much using at the moment, until we can find something better.
Has there been any thought about what issues, if any, there might be about putting amoxicillin into the ocean for other species, fish or, other corals? Is there any information or science on that?
So yeah, I could give an answer for that. So in the presentation, I mentioned that there was going to be a study specifically looking at the effects of amoxicillin from the treatments in areas on coral reefs where there are fish and life. I guess once that study is concluded, I'm not sure if anyone else is doing that at the same time. But one thing that we do know is there have been studies where they've noted that there haven't been any changes in the surrounding immediate environment to other corals, like any other diseases popping up or any adverse effects on the fish or anything, or the corals, just from the surrounding areas. And they did that over the course of a year and didn't find any significant changes in any abnormalities or things like that. So things could arise in the future, but this has been looked at, and obviously it's a major thing to point out. Obviously in large quantities that there may well be an effect of antibiotics in the marine environment, but as we know right now, it seems to be okay, and it's quite localized within that coral as well, and it's hardly leaking out unless a fish is tearing at it. Yeah.
The other question I had was, you were wearing gloves when you applied the paste to the coral. Are those gloves that you guys provided for us? And the other question was I right in seeing that you basically put the paste on your actual fingers to apply it with your fingers rather than use the sort of syringe thing to apply the paste with?
Right. So the first answer is, yeah, we do provide the gloves for you. The second answer to your question is, we don't put the treatment onto our hands and then apply it. We literally put the syringe up against the coral on the lesion, and then we dispense it out by pressing. It's hard for me to show you here, but as you imagine me in that video, we're literally putting the tip or the nib of the syringe on that disease boundary and pushing the treatment out just to make a line like Play-Doh. Yeah, we only use our finger to smooth it out across the boundary, so we get a nice spread amongst the disease boundary and the healthy boundary, but not so much the healthy because we don't want to kill too much. But we just use our finger just to even it out and make sure it's adhered to the exposed coral skeleton that's recently diseased.
And then my final question was, you showed a slide where I think it suggested that 80% of the corals were in an okay situation after a period of time. Could you just go through that slide again one more time? It was kind of like a ramping up series of red bars. I just wasn't sure what the conclusion there was.
Let me I've closed the presentation. Let me just open the presentation again and I'll go back to it.
Sure.
Yeah, so, okay. So yeah, we're just basically performing an experiment ourselves just to assess the effectiveness of the treatments. So in this graph, just basically what you're seeing is in June, this is where we started tagging each of the diseased corals. Now they were diseased already, and in our first day we treated them and then marked down that we treated them. And then over time, we've just been seeing that less and less of the corals actually need like retreating. So we think that for us periodically revisiting these specific corals that have been infected and treating them, that over time, they're just becoming more like healthy over time. They're just responding quite well. And, they're not getting as many new lesions as they did in the earlier stages of the infection. So that's just like promising news to us. And, we do think like over time, as long as you keep going back and applying the treatments to corals, that, we can save them. And that's our mission.
And if I recall, you said you did this sort of weekly, and is that right? And then the other question is, I could read it now. I've switched machines, but
Okay.
you're on, you said the corals are stable. So I assume that just means that whatever damage that occurred from SCTLD originally just is retarded and is, I don't know, if 20% of the area on a coral had been destroyed because of SCTLD, the other 80% just stays there, or maybe there's some small ingress through time, but effectively you've stopped it. Is that kind of what that means?
Yeah, thanks for bringing that up. So yeah, we visit this site on a weekly basis every Friday, if you want to know. And yeah, that's a good point bringing up about the stability. So what I mean by this is that I'm classing a coral as stable if it hasn't been treated for at least a month from its last treatment. I could change that boundary to 2 months, but I just think a month is a good time where, we've treated it and then it's been a month and we haven't seen any new lesions, or we haven't seen the disease progress past that treated lesion, if you know what I mean. So yeah, that 20% that's already dead, that's gone. But then that 80% has been uninfected for at least a month, and then I'll mark it as stable. But then this can be reset back to 0, like when they get a new lesion or something else happens. Yeah.
Thank you. That's great.
No worries.
Sorry, Jay. So did you say the name of the brand or format that you get the amoxicillin?
It's so there was one that we were using called it was from a company called Phytotech Lab, which is based in the US. And there isn't like a particular brand of antibiotic that they are selling. It's just from them. But I imagine they just get their antibiotic through other various sources as well, and just they rebrand that as Phytotech Lab. But we think it's essentially the same. But because we have switched antibiotic from Phytotech to a wholesaler where we've cut out the middleman, we're going to be monitoring the treatments with the new antibiotic that we are using. Like, it's still amoxicillin, and we think it's got the same properties as the one from the US that we did used to buy. But yeah, we think it's essentially the same purity and everything, but we're going to monitor how well the corals react to this amoxicillin that we got from India. Does that answer your question?
Yeah. Would it be possible to put your email address in the chat as well?
Yeah,
Copy
sure.
in.
Yeah. Okay, there's my personal email. Should anyone have any more questions about antibiotics or any aspect of the SCTLD project, they can reach out to me or Croy, who's in charge of the project. Like we're happy to help. Has anybody got any further questions or?
Thank you so much.
Yeah. All right. No worries.
And then you guys will be in touch about the field dates.
Sure. Yeah. So I think there's another training session tomorrow.
Mhm.
Yeah.
And then after that, we'll be in touch. We'll add you to a WhatsApp group with everyone's contacts in there, and we'll put out a schedule. We'll ask when we need people to come in, and we'll see who's available, and we'll go from there.
Brilliant. Justin just posted a message in the chat.
Okay. Has there been any weekly? Recolonization of what? Of the coral itself or something else?
Are you referring to new larval settlement?
Yes, of corals. Well, as it's so early in the stage of the project where we are monitoring the effectiveness of the antibiotics on the diseased and dead lesions, well, areas, we do get the algal growth, but then this doesn't mean that the coral can't then grow over dead areas of itself. So it just basically like resheets, and it can basically grow on dead areas of that coral, if that makes sense. But we haven't seen anything yet. It's too early. It'll be like a year or so.
I mean, as Jay mentioned, resheeting is over time. It's not something that happens within a few months or a year. And, my experience on coral reefs is occasionally we do get new settlement of larvae over time, but you got to wait for spawning to occur. But on large colonies, in the past have shown where they do resheet once the disease stuff has stopped, and they open real estate for it to do that rather than being colonized by macroalgae and such. But it doesn't happen right away.
No.
These things live 500 years. We are just a speck in their lifespan. And I think that's one of the things that as humans, we don't put into context, the evolutionary history of corals. And that's why it's alarming that we see such loss in our lifespan versus, their lifespan is hundreds of years. And they regenerate themselves by consistently laying down, layers of calcium carbonate and new growth. And it's a continuous life like that. And then something like stony coral tissue loss comes along and just puts a total, stop to that process that's been going on for hundreds and if not millions of years. So it should concern everyone.
So I think my microphone wasn't working before, but part of the concern that I had with that was, with this being suspected to be viral, if viral particles are able to basically remain on the dead areas, would that potentially prevent coral sheeting or recolonization? And has there been any study that you're aware of that's been done into that?
There's I'll take this, Jay, sorry.
Yes.
There's
sure.
multiple and many studies going on trying to wrap our heads around this whole, tissue loss. They've been in the past where have been shutdown syndromes and different things that have caused similar responses, physiological response, but nothing that is persistent and is rapid and continues on for year in, year out, through no seasonality to it. But the more recent, the sort of breakthrough that we have, or understanding of it, is that it's a long filamentous single-strand RNA, very similar to the morphology of a plant virus. Now, saying that, as I understand from the report, is the fact that it suggests a breakdown of the host cell and the zooxanthellae symbiotic relationship there. And it's not very clear which happens first, whether it's the host or the zooxanthellae. So that being said, it points towards a virus, but it could also be a bacterial pathogen coming into play in the whole thing yet. So until we can, we're so behind in our limited knowledge of coral diseases, until we probe deeper and further and do our homework trying to understand what's actually going on. Right now, we only know it's partially viral based, but it could also be a pathogen coming into play, or both at the same time. So
Thank you, Corey.
Any other questions? Yeah. All right then. Well, once again, thank you everyone for participating in the training. We look forward to seeing you tomorrow. Unfortunately, we will be taking that. It'll be 2 other people, but enjoy that, and then hopefully in the very near future, you will be coming out with us. Yeah, have a lovely evening, and good to see everyone.
Also, if anyone have any deeper questions that they would like to ask, feel free to drop me an email. I just put my email on the chat there, so be more than happy to try to answer them for you.
Thanks,
So,
guys.
okay, well, thanks again. Have a lovely day.
Thank you. Bye now.